David Baltimore, Robert Andrews Millikan Professor of Biology at Caltech. The HIV epidemic has been one of the most disruptive emerging virus infections that this world has ever seen. I first heard about the AIDS epidemic even before HIV was discovered. Once the virus was discovered to be a retrovirus, and that's the kind of virus that I've worked with now for many years, it seemed to me it was a responsibility for people who worked with retroviruses to use their skills to study HIV. I was looking at the HIV field years ago and realized that standard vaccination methods were not doing very well for HIV. I didn't know whether one day somebody would find a way to use vaccination methods, but I did know that the field needed people to think differently about the problem. Rather than trying to use a virus as a vaccine, we decided to take antibodies that had already developed and see if we could make people produce those antibodies as protection. During the time we were trying to find ways of using existing antibodies as protective measures, Alex Balazs came to the lab to help us with this. He had a tremendous background in understanding vectored production of proteins.
What we call VIP stands for vectored immunoprophylaxis. It's a process in which we use a viral vector carrying the genes for an antibody, in this case one that neutralizes HIV, and we use this modified virus to infect the muscle cells of an animal, and this produces antibody into the circulation that is then present to neutralize virus when you're exposed to it. So what we found was that we could deliver broadly neutralizing antibodies to mice, and that by doing so, mice would produce a lot of antibody, and that antibody was then able to prevent infection with HIV. We were surprised by how well it worked, actually. We thought this might give some protection, but we were very surprised that we were actually able to challenge mice with very large amounts of HIV and have this antibody neutralize and protect the animals.
I was gratified by the results of this study, but I wasn't surprised in the sense that we knew these antibodies were effective, and once we had learned that we could make them at high concentration, or have animals make them at high concentration, then I was expecting that it would prevent infection, and of course it did. HIV research has generally been a series of negative surprises. The virus is not sensitive to standard vaccination. The virus is not easily controlled. This is at least a positive step forward. Whether it becomes a human prophylaxis or not will depend on what we find when we go into humans, both in terms of safety and in terms of efficacy.
For me, it's just been great to work with David. Obviously, he's well known and well respected, but it's deserved, very well deserved. And David gives you the freedom to really just take whatever idea you have and run with it and see what'll happen.
The next step for us is to find out whether humans behave like mice. Now, the history of vaccine research is that humans don't behave like mice. And so we have to be prepared for surprises in carrying this work to humans, and we may have to go back to the laboratory and solve new problems that we didn't have when we were dealing simply with mice.
Assuming all of the clinical safety trials progress without issue, we would be very hopeful that this could represent a truly effective HIV vaccine.
The advantage of VIP over standard vaccination methods is simply that it works. Right now, I think it's the best thing on the horizon.